CRISPR is finally here. I'm so excited about this. It has recently been approved by the FDA for the treatment of sickle cell disease and beta thalassemia, which are two of the most devastating diseases affecting millions around the world. This is Johnny. Johnny has been in and out of hospitals his entire life. He's been dealing with extreme pain and suffering at such a young age. But luckily, Johnny is one of the first patients in the world to receive this special CRISPR treatment for sickle cell anemia. And it's working. Johnny is doing really well now. This treatment, called Casgevy, have just been approved by the FDA and its UK equivalent for the treatment of sickle cell diseases and beta thalassemia. So today, I want to talk about the science and the story behind this treatment. But I also want to talk about the dangers and the limitations of this treatment. Genetic hereditary diseases such as cancer, cystic fibrosis, and even sickle cell anemia are caused by errors in our DNA. DNA has the blueprint for everything that happens in our cells, our organs, and in our body. The cells use DNA to produce RNA, which they use to produce proteins. And the cells use proteins to do pretty much everything. So if you have a mistake in your DNA, you're going to have a mistake in your proteins. And this mistake is going to turn into problems in your cells, organs, and your entire body. This is where genetic diseases come from. And since we inherit DNA from our parents, there's a chance that the mistake is going to pass down from one generation to another. The idea of gene therapy goes like this. If we know there is a mistake in our DNA that's causing a disease, and we know exactly where that mistake is, why not go into our DNA and fix that mistake? Because if you fix the mistake, the disease should go away, right? Right? Well, not really. It's not as easy as it sounds. It turned out to be one of the hardest scientific questions of the last few decades. Scientists have tried a lot of methods for gene therapy, but many of them did not work. The CRISPR-Cas9 technology was a groundbreaking gene editing technology introduced in 2012. It was proposed by Dr. Jennifer Doudna and Emmanuel Carpentier. And since then, it has revolutionized our approach to genetic engineering. In 2020, the two scientists received the Nobel Prize in Chemistry. And just 11 years after its introduction, it's already being used in clinical trials and hospitals. The CRISPR technology uses a protein called Cas9. Cas9 is a protein used by bacteria as a defense mechanism against viruses. And this is how it works. It can recognize a specific segment out of a long sequence of DNA. This is how it can recognize specific viruses to destroy them. So since this system was very specific and efficient, these two scientists got a brilliant idea of using it to target human DNA. And this is how they did it. First, you have to know your target sequence. For example, a mutation that's causing a certain disease. Then you design an RNA sequence tailored to match your target DNA. This is called guide RNA. Why is it guide RNA? Because it's going to guide your Cas9 protein to the specific DNA location in the cell. And once it gets to the location, it's going to act as scissors and cut the DNA at that specific location. And after the cut, you have a few options. You can remove the DNA segment completely or replace it with another DNA segment. And after this, the cell naturally repairs the break using its enzymes. Now back to sickle cell. It is a genetic disorder that affects hemoglobin. Hemoglobin is a protein in red blood cells that carries oxygen to different parts of the body. In the sickle cell disease, there's just one single mutation in the genetic sequence of hemoglobin. If you look at the sequence, A, which is adenine at position 17, is replaced by T, which is thymine, that is 8. This single change leads to an abnormal hemoglobin protein, and this altered hemoglobin causes cells to warp into a crescent or sickle shape. These sickle-shaped cells tend to clump together, causing obstructions in the blood flow. And this causes complications in the body such as severe pain, organ damage, and a shortened lifespan. Victoria Gray, a 34-year-old sickle cell patient, described it as sharp pain all over the body. It is deep pain and you can't touch it to make it better. That sounds really painful. This is a serious global health disease, and it seems to disproportionately affect people of African descent. And unfortunately, we don't have a cure for it yet. We just have medicine to take care of symptoms and expensive treatments such as blood transfusions and bone marrow transplants. So this is a disease affecting 10 million people per year, and it is very expensive to treat, especially in places where it's more prevalent. So you can imagine how scientists have been looking for alternative treatments for years. These two companies, CRISPR Therapeutics and Vertex Pharmaceuticals, wanted to use CRISPR to not only treat sickle cell anemia, but another related disease called beta thalassemia. So this is what they did, which I think is very clever. They did not target the sickle cell hemoglobin itself. They found another way around. They targeted this protein called BCLA11A. Why? Because this is a protein that stops the production of fetal hemoglobin. Now, fetal hemoglobin is amazing. It's actually better than adult hemoglobin. But when you become an adult, this protein stops the production of fetal hemoglobin. So if we remove this gene, we're going to start producing a lot of fetal hemoglobin. And blood cells with this hemoglobin are going to be very efficient at transporting oxygen. With the fetal hemoglobin, we're also going to have more of these healthy, efficient cells and fewer of the sickle cells. So this is how the treatment goes. They remove some stem blood cells out of the bone marrow of the patient. Then they treat the cells outside the patient in the lab. This is where CRISPR is performed. And after you perform CRISPR on the cells, you inject the transformed blood cells back into the patients so that they can start producing the fetal hemoglobin and healthy blood cells. As you can imagine, it is a long procedure, but these two companies were able to successfully do it and basically cure sickle cell for over a year. Victoria Gray, the patient that we talked about earlier, was the first patient to get this treatment back in 2019. And she's now doing well. All of her symptoms have basically disappeared. In the UK clinical trial, the treatment worked in 28 out of 29 patients. This is a 97% success rate. So this treatment is promising and it's working really well so far with minimal side effects. Dr. Doudna, the pioneer of this technology, says that she's excited and a bit overwhelmed with emotions at the news of the approval of this drug. She came up with the idea just 11 years ago. This is exciting news and the future is promising. But we have to talk about some of the potential dangers and limitations of this treatment. First of all, it is not a complete cure. It is not directly targeting the sickle cell hemoglobin. It produces enough fetal hemoglobin to alleviate the symptoms of sickle cell anemia, but the body still has the ability to produce sickle cells. So after going through this year-long arduous treatment, these patients might have to do it again in the future. Another serious consideration is that whenever you change a gene like this, you have to be conscious of unintended alterations of DNA elsewhere in the cell. We've seen this before in gene therapy treatments. If you change a gene by accident, consequences can be severe depending on what gene you're changing. So we have to be very careful, test the patients regularly, and wait and monitor long-term effects of this treatment. Another drawback of this study is that it is so expensive. I want you to guess how much you think this treatment costs, and I promise you that your guess will be wrong. You want to know how much it costs? $2 million. That is a lot of money. Let me know if your guess was anywhere close to that. And as a reminder, sickle cell anemia affects a lot of people of African descent. And most of these people are in the sub-Saharan region of Africa. So this is not going to work unless we dramatically decrease the price, especially if we might need more than one treatment. Now, our lab works on different approaches to target cancer. And I can't wait to see the CRISPR technology applied successfully to cancer. We have a few clinical trials for CRISPR treating forms of leukemia, carcinoma, and lymphoma. We even have CRISPR clinical trials for HIV and diabetes. So this is very exciting, and I'll definitely be reporting on future progress of CRISPR technology. So if you're interested, subscribe and stay tuned for that. And if you want to see a video where I talk about another promising cancer cure, click the video right here, and I'll see you in the next one. Bye-bye.